Thank you.
A research scientist with appointments at Picower Institute of Learning and Memory at MIT, an instructor in radiology, Harvard Medical School, assistant neuroscientist at the Martinos Cetre for Biomedical Imaging, Massachusetts General Hospital, and the Broad Institute in Cambridge, Massachusetts. He has developed sensors for brain pressure measurement in newborn babies and an artificial arm prosthesis to below elbow amputees during his doctoral work at the IIT. As a postdoctoral fellow at MIT, he studied brain plasticity by rewiring visual and auditory circuits of ferrets to show the influence of environmental inputs in shaping and developing brain circuits. At present, he is studying the relationship of genes, circuits, and behavior to uncover brain mechanisms of autism, and his work has appeared in top scientific journals including Nature, Science, and Neuron. We would like to welcome Jitendra Sharma to talk about Between Genes and Behavior, the Circuitry of Autism.
Hello everyone. I'm so glad to be here because this is such a wonderful conference. And I must thank and congratulate Mr. Raghunathan and his wonderful team who put together this amazing program. And there's a lot that I've learned. And I'm going to tell you something slightly different from what we have been hearing. This is about the connection between genes and behavior, which is the circuitry of autism. And in this talk, I'll try to convince and probably compel you to recognize that there is something missing in our understanding of genes and behavior. We have to understand the circuitry of the autism, the circuitry of the brain as a whole, if we want to understand autism and how to treat it. World over, the incidence of brain disorders and brain diseases has been increasing rapidly. By some accounts, we expect that the incidence of diseases of brain and brain disorders will increase and outstrip that of cancer and heart diseases very soon. That's a lot of burden, and something has to be done about it. But we have not had much progress in terms of alleviating this, actually treating the core symptoms of these diseases, and we must find new sciences that will help us how to tackle this. So autism and other neurodevelopmental disorders are developmental in nature, which means that they develop over time. By the time a child is about 2 to 3 years of age, he starts showing atypical symptoms of behavior. I need not go into it because it has been talked about so much. But the parents get concerned and they take the child to a doctor or a psychiatrist who runs a battery of tests, sometimes genetic tests also. And if the scores match up with the symptoms in standard criteria, then the child is put on a spectrum. And then so on and so forth. The therapies and occupational therapies and behavioral therapies go on. We have known for a long time that autism is a polygenic disease or disorder. Multiple genes are affected, and we have a fairly good idea about where these genes are located and what do they do. We also know a lot about the behaviors, the atypical behaviors, the behaviors that a child starts to show very early. And we can systematically also fine-tune our strategies of finding behaviors and their patterns and accordingly institute therapies. But there is still something missing. The missing link is that genes actually create circuits, and circuits have function, and those functions determine behavior. We must understand this link if we really want to get ahead in this problem. So brain has different circuits which are dedicated to different functions. And some of these circuits, Shiva Arnold yesterday talked about some of those circuits which go awry in autism, which are related to symptoms which you can see. But this thought of brain circuits, that brain is a giant circuit. It is one of the most complicated circuits that we know. Is not very new. Scientists have been tracing morphology of different types of neurons for over a century now. But it was only in early 90s that people started to wake up that brain is actually a giant circuit. There was this landmark paper, the picture that I'm showing you is from Phelan and Van Essen, which has been shown hundreds, maybe thousands of times by many people. What it showed is the circuits in the brain that process visual information in a monkey brain. They painstakingly traced these neural circuits and showed to the world for the first time that as many as 32 and even 38 areas in the brain process some aspect of vision. Vision starts from the eyes, but then it courses to the back of our brains where the primary visual cortex is. And from there, the information is sifted through, it is processed in different ways and integrated, and it creates complex behaviors. Fast forward 30 years later, one of my colleagues at Harvard, Van Wideen, he used diffusion spectrum imaging, a functional MRI technique, to trace the circuits of the axonal tracts of the entire brain in a 3D space of a human being. These colors basically show the traces where they are, the tracks where they are originating from and where they are going to. What I want to show you is the complexity there, and this is still not complete. A couple of years later, in 2015, Bobby Kasturi, who was a postdoc in Jeff Lichtman's lab in Harvard, he took a small speck of the part of the brain of a mouse, about 200 micron thick, which is about 100th of the size of a grain of sand. And he sliced it very finely, used very sophisticated electron microscopy and computer algorithms to reconstruct the circuit. And what you see here is a jungle of wires as if. They were able to connect every part of this circuit to every other part. And it just shows the complexity of the problem that we are looking at. By their account, there are actually quadrillion connections in a mouse brain. How do we tackle this problem? There's something more than that. Just tracing these circuits is one part, but understanding what these circuits are doing, what is the function of these circuits, and how do they get specialized in doing specific things, which creates complex behaviors, is the key thing. So my own understanding of circuits in the brain also came about in early 90s. It was a coincidence and a happy coincidence that actually changed my life, literally. I had finished my PhD from IIT Delhi and All India Institute of Medical Sciences, where I was making sensors for brain pressure measurement non-invasively in newborn babies. And on the side, I was also devising different aids for the handicapped, including a prosthesis for hand amputees. I had finished my PhD and somehow I got interested in going to the government, so I accepted a central government job, which was a very good job. And I did very well. In 5 years, I got 3 promotions. And it was a cushy life, but there were endless meetings, endless cups of tea, and making plans for the national stage was my bread and butter. Then suddenly, one fine day, my PhD advisor, he called me that, Sharma, there is a talk from a professor from MIT, and he is going to talk about rewiring circuits in the brain, and you should be interested. I was totally struck by this rewiring the circuit. What? Can you rewire the circuits? Brain is not a circuit. There are no wires there that you put them and you light a bulb and then you change those wires somewhere else and light some other bulbs. It cannot be that simple. So with intrigue and some fascination, I went for the talk. And that was the most amazing talk I ever heard. Professor Mridang Sur from MIT was actually talking about rewiring the brain in a small ferret, a ferret which was 1 day old. They used very clever microsurgical techniques to put the inputs that are going from the retina, from our eyes, and somehow reroute them so that they are, instead of going to their home, which is supposed to be primary visual cortex, they put them into the auditory area where we hear from. And then they recorded from the auditory area to find out whether single neurons there are responding to vision or to audition. And they found that some of the cells indeed were processing visual information. So here was something very different that people had known before that. After the talk, I went to Professor Sur and asked him, can I join you? But before that, I also asked him, where you are, is this story fully complete or there's more to be done? He said, no, we just scratched the surface. So I asked him, can I join you? He said, what do you do? I said, I'm a government servant. I push papers. He said, well, there's a fellowship. If you win this, you can join my group. It was a very competitive fellowship. And I'm the last Indian to get it. It's the Fogarty Fellowship of NIH, which they stopped giving to Indians now. But anyways, after 1 year, through a stiff competition, I got this fellowship, and I was at MIT, a postdoc. I left my cushy life of a senior government official and went there. And I launched myself head-on in this rewiring work. Very soon, after 2 years, I was the leader of the group, and we were doing very complicated experiments, which would last typically about 3 days. I would do 2 of those experiments every week. And after 6 years of backbreaking work, we ourselves published a landmark paper in Nature, in which we indeed showed that not only these ferrets, when we rewire them, they have single neurons or clusters of neurons that are responding to the vision, but the entire machinery underlying that has also changed. We create a new function in a new part of the brain. We did sophisticated mapping of the brain areas. We also did multiple electrode recordings and all. The upshot of that was that we were able to show that brain has re-specified itself. The inputs have changed the brain. Not only that, we also trained these animals in a water-based task in which they were allowed to go for sound or light. And what we saw was that these animals, the rewired animals, were indeed going for light, which meant that not only we changed the circuit, we also changed their behavior. They were using these circuits for their own behavior. So that was a story that changed my life. This is another aspect of that study where we recorded from different parts of this circuit and found that there are neurons that are responding to vision. So going back to autism, how do I relate this? We know that autism is a polygenic disease. There are more than 1,000 genes which are now implicated in some aspect of autism. And our key question was, how do I relate the genes with circuits? We cannot tackle all these 1,000 genes, but there are some monogenic disorders also. You must have heard of Fragile X syndrome, in which FMR1 gene is affected. Similarly, Rett syndrome, which is affected by MECP2 gene, and so on and so forth. But there is an interesting gene that we were interested in because we wanted to understand circuits. And we were looking at a gene that actually is involved in making circuits. And that gene is actually a SHANK3 gene. A SHANK3 disorder, or also called Phelan Mcdermid syndrome, has children who have this gene mutation, and they have some very specific facial features. This gene lies on chromosome 22, which is, and it is on the long arm of this chromosome. As you can see this is a chromosome 22, and the yellow arrow that shows the location of this gene. So we know precisely where this gene is located and what does it do. It does something very interesting. So in the brain, how circuits are made. There are cell bodies. They have an input which comes from the dendrites, and then there's an output fiber, which is called axon, that takes information, and then there are junctions, which are called synapses, where this information is transferred from one cell to another. And this gene, as it is shown in the blue, this is the structure of the synapse, the most critical element of the circuit. This gene is involved in making this synapse. It is also involved in the transmission of the information from one synapse to another. So this is a good gene because it is directly involved in the circuit making. So in about 2016, a friend of mine, who is also a very creative scientist, Yang Zhao, in Goping Fang's lab at MIT, he created a mouse model of this gene deletion. And he studied extensively this gene. And he found that indeed, there was a problem with the synapses of the circuits. He also tested behavior in these mice. And he also found that there were some behaviors which could be human-like in BMS patients. And there is a lot that we could learn from that. But of course, mice are mice. They don't have behaviors as complex as human beings. But they do have a brain machinery which is almost similar to humans. The only difference is that our brains are more expanded. But later on, we also found over the years that there is something which is specialized in human and monkey brains, which is we are very visual animals. About 70% of our brain machinery is dedicated to processing vision. So the areas that process vision and make some sense out of it, they are expanded in humans, whereas mice have something else that is expanded. So we realized that we need a bridge animal or multiple model animals in between if we want to really understand what is going on, how these circuits are different, and these circuits are doing different computations. So in about 4 years back, a decision was made at MIT that we thought that we will create a primate model of autism using this gene. We were also very fortunate that around the same time, a new technique of gene editing was coming up. Actually, a collaborator and a colleague of mine at MIT and Harvard, Feng Zhang, was leading the pack. The technique is very simple. We know that DNA is a code of letters, or genetic code. And genes make specific patterns which are on this DNA. What we need to do is, if we want to change any part of the gene or delete or add a gene, we need to know where precisely it is located and to make a template of it. And also somehow create a way to guide this template to the specific part of the gene. We also need to have something to cut and paste this DNA, because if we want to introduce a small template, we need to cut, we need to introduce, or we need to delete. We have to rejoin things. Interestingly, this aspect of gene repair is going on in our system all the time. Our DNA is all the time getting changed, and there's a repair and a mechanism which is going on all the time. So there is an enzyme, there's a molecular scissors which is available in the system. Once we recognize that, then what scientists do is they create these templates of the genes that they want to manipulate, and they mix it with this enzyme or this molecular scissors and inject this construct in a developing embryo of a fertilized monkey or mouse or human being. It can be done in any fertilized embryo. So since we chose primates, so basically you have a primate, you take some DNA out of it by taking some skin cells or blood, and you have a DNA of that. Then you create a template of the gene that you want to delete or introduce. And then you mix it with the molecular scissors, which is this Cas9, and then you put it in the embryo. It has to be done at a precise time when the embryo is developing and it is divided into cells. So the timing is the critical thing. And then what Yang Zhao did, he put this guide RNA, which deleted this gene, which is called SHANK3, and we injected this We did in vitro fertilization, and in a receptive female, this new mutated embryo was implanted. It's a standard in vitro fertilization. And monkeys have a gestation period of about seven months. Humans have 9 months, monkeys have 7 months, at least these kind of monkeys. And we waited, and we got pregnancies. After 7 months, 5 newborns were born, which were tested extensively by us.